Hello, how are you everyone? This is Yapaksa Tuition Center and welcome to biology lesson prepared by Mr. Yapaksa. So what are we going to look at in this context? This morning we are going to look at germination. We are going to define what germination is. We are going to look at how a seed germinates, what happens during germination, and we are going to end with two types of germination. Germination is the word. What do you think to your mind when the word germination? So to germinate just means a seed has to grow from a smaller seed to a seedling. So in this case, we may have a definition of clear picture like is the process by which seed begin to grow and develop into a new plant, which is a seedling from an embryo. Or there are other definition you can use that can still connect to this one. Condition for a seed to germinate. So for a seed to germinate, it will need a lot of conditions. So in this case, we have one, water, oxygen. I'm sure you can guess the last one. Correct. You get it right. Warmth or temperature. These three conditions are needed for a seed to germinate. Without this, a seed cannot germinate. But what happens during germination? Since now we've talked about those three conditions, these are the other things that happen. Seed absorb water. Seed coat break open. Embryos start to germinate. Roots and shoots appear. After these ones, now they've shown that interest that now they're germinating. Another important result also happen. What is that result? Now, for the next content, let us now go into details. We explain more about germination. Meaning in this case, we're going to look at the type of germination. So the first type of germination is the epigal germination. What is epigal germination? This is the type of germination where the cotyledons are pushed above the soil. You can see on my diagram side here. Let me just come here with a blue pen. This seed, then this one is a hypocotyl. Then the cotyledon here, it is pushed above the soil, meaning the soil here is mainly downwards, but it has been pushed above the soil. So that's what we mean by saying cotyledons are pushed above soil. So cotyledons are pushed above the soil. It's now how it works. Let us dive a bit into another content or context. Now in this case, what are we going to talk about? This happens because the hypocotyl grows longer and lifts the seed leaves upwards. So this diagram represents hypogel in beans. Examples of these, we can talk about beans, sunflower, and cotton as well. Then the other type of germination here is the hypogel germination. Now the cotyledons remain below the soil surface. This happens because the hypocotyl elongate instead of the apocotyls. Example, we have maize and peas. Then this is the diagram. We need now to talk about the differences between the two, which we've already explained in the other content. So now these you can just happen to see, then you can get some ideas. Then you end with the last diagram for germination. Thank you.